I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. I'm your host, Jesse Lawler, excited to bring you the 56th episode in this podcast dedicated to the betterment of your own brain by any and all means at your disposal. This episode is going to be a little bit of a departure from smart drugs and nootropics stuff, but it's definitely going to be very medical. It's going to be dealing with brains and our entire bodies. Our guest is going to be Dr. Thomas Seyfried of the Boston College, who in 2012 published a book proposing that nemesis of humanity, cancer, is actually a metabolic disease rather than a genetic disease. We'll get into what that means in the course of the interview, but the real-world takeaways from that idea are pretty profound, and I wanted to have him on. I heard him interviewed one or two places around the internet, and the ideas sounded really fascinating, but there was a couple questions that I really wanted answered that hadn't made it into the other interviews, so I figured I was going to give him a call and get him on this show. So we'll be talking with him in just a couple of minutes. If you hang around until the end of the episode, I'm going to tell you which Christmas time seasonal favorite food might have some real health dangers. So stay tuned for that. And in the meantime, let's do This Week in Neuroscience.
Smart Drug Smarts. This Week in Neuroscience.
Okay, so this is pretty far out stuff. About a month ago in Nature Communications magazine, there was an article called Mind-Controlled Transgene Expression by a Wireless-Powered Optogenetic Designer Cell Implant. What the hell does that mean? Well, the short version of it is this. Okay, so let's think about genes and the way that we've thought about genes over the course of history. At first it was that you get your genes from your parents and they're fixed and you're sort of, blessed or cursed with whatever your genetic heritage is, and that's it. What you're born with is what you're left with. What we've learned really in the past 10, 15 years is that there's a huge amount of variation in gene expression throughout an organism's lifetime based on something called epigenetics, which is basically a subcellular response to environmental factors within the body that control which genes are actually expressed to create proteins. So just having a gene doesn't necessarily bless or curse you anymore. It's really about whether that gene is in an on state or an off state as far as whether it's going to be producing the proteins that it codes for. Now, this article is kind of about the next weirdest step. So what if instead of just environmental factors like, I ate some paprika and paprika makes these genes express, what if you could control the expression of genes with your thoughts, with your mind? That'd be pretty weird, freaky sci-fi stuff, right? Well, that is basically what scientists have done now. They say that this is about 10 years away from being usable by humans, but the short version is they created a device within a mouse. They implanted this device into mice that has some cells in it that are sort of hidden behind a membrane, but these cells are producing specific proteins, which are then released into the mouse bloodstream. The proteins that are produced by these cells in this sort of interorganism gizmo are being controlled by light coming from a little LED that's in the same gizmo. Now, this gizmo's LEDs are turning on or off depending on the brain waves of humans that are involved in this experiment. So they've got some humans that are hooked up to EEGs measuring their brain waves. So they're either getting in a super focused state, like playing, I think they said Minesweeper was the game in question, or a meditative, super relaxed state. And these brain states have different frequency signatures that are picked up by the EEGs. These are transmitted over a Bluetooth signal to the little gizmo inside the mouse. The gizmo inside the mouse turns on or turns off LEDs, so they're flashing in certain ways that activate these light-sensitive proteins also within the gizmo. And these light-sensitive proteins are in turn epigenetic on-off switches for which proteins are going to get created within this same little gizmo and then released into the mouse bloodstream. I hope that kind of made sense, but this is like a duct tape sort of together thing. Obviously, they didn't want to test this first inside humans, but they've got human volunteers putting their brains into a certain state, which is in turn activating gene expression within a little cellular gizmo within a mouse. So if you kind of remove some of the steps from that process, if you, for example, had this gizmo not within a mouse, but within that same human, in theory, what this kind of thing could lead to would be somebody's brain state being able to have direct epigenetic effects within their own body and the release of certain proteins that would have effects on their own biology and metabolism. And that is pretty far out. This work was done by researchers at MIT and ETH Zurich. And needless to say, we will put a link to this up on the website.
Smart Drug Smarts.
In the audience interaction department, got an email this past week from a listener named Jack in Australia who said, just wanted to send a big thank you for your latest podcast with Con Stow. I'm from Melbourne. Actually, he would have said Melbourne. I'm from Melbourne, Australia, and have been trying to decide on which university to attend to focus on psychology and neuroscience. Con Stow's research and department at Swinburne is exactly what I was looking for, so I just wanted to say thank you for showcasing Con's research. You've made my university decision a whole lot easier. That's super awesome. We love getting emails like this and knowing that our humble little podcast is having an actual effect on real people's lives. Thank you so much, Jack, and yeah, best of luck there. For those of you that has no idea what he's talking about, he's referring to last week's episode, which was Professor Con Stow talking about plant-based cognitive enhancers, nutraceuticals, things like that. Another couple of pieces of cleanup news from some old episodes. We had the oxaloacetate episode a few weeks ago with Alan Cash, and he was talking about oxaloacetate and its ability to help the body sort of get into a state that mimics caloric restriction and some of the life extension benefits that might go along with that. As I mentioned, I realized after the interview that I forgot to actually ask him the million-dollar question, which is, how his use of oxaloacetate actually benefited his own personal biochemistry. He emailed me back and he said that he's 57 years old and his fasting blood glucose levels are 81 through 83, which is indeed quite good. So for those of you that were scratching your head about that open loop, now that is closed. And also, in the realm of things that are somewhat annoying, but there's nothing I can really do about, we did a switchover on our website this past week. We kind of moved up to a new theme for the look of the site, but in the process of having switched over, we lost a couple of listener comments that had come in during the time when we kind of like divided off our staging area site from our at the time live site, and some of those comments didn't make it across. So I really apologize if anybody listening is one of the commenters. I've got the email record, but not everybody that leaves a comment on the website has their email address associated with it, so I didn't have a chance to get those comments from a couple of previous episodes published on the site nor responded to. And if you're one of those people that's fallen between the cracks, we're not trying to be jerks and ignore you. It's just one of these technical glitches that happens every now and then during the process of a site upgrade. That said, we do have a kind of cool new look for the site, and it's going to be getting even cooler soon as we add some new stuff. So check it out if you haven't been by smartdrugsmarts.com in a while. And I will also put in the obligatory plug if you are not signed up for our mailing list. Please do, so we can let you know about cool new stuff, sometimes even before, sometimes even in addition to the things that we mention on this podcast.
Smart Drug Smarts, where we turn information into sound, into bits, into packet data that turns back into bits and sound, and then into neurotransmitters that release funds that release mail-order synthetic chemicals that cross the blood-brain barrier to release augmented performance from your brain.
So Thomas N. Seyfried is a professor of biology at Boston College. He got his PhD at the University of Illinois, Urbana-Champaign. His fields of interest include gene-environmental interactions, lipid biochemistry, neurodegeneration, epilepsy, brain tumors, ketogenic diets, dietary energy restriction, and all of these have swirled together over the course of his career to give him apparently some fairly unique insight into how cancer might be treated and controlled. He published a book in 2012 with the title Cancer as a Metabolic Disease on the Origin, Management, and Prevention of Cancer, which is available for the bargain price of $120 in hardcover and $80 on Kindle for you Amazon buyers out there. Bit of an expensive one, but we're going to be covering a lot of the high points in this coming interview. Probably almost everybody listening knows somebody or has known somebody who has had cancer. I certainly have known a couple of people in my life, both young and old, who have dealt with cancer. And it's something that while nobody likes to think about, probably everybody should be thinking about because it's such a major killer in our world today. So even though this is a little off topic from the normal subject matter that we're covering, I thought this was something that would probably be interesting to everybody because of what seemed to me like a fairly profoundly different way of looking at and treating cancer versus what I'd been aware of in the standard way of doing things. So with that as preamble, Dr. Thomas Seyfried.
Ketosis is linked not only to cancer and brain health, Alzheimer's disease, Parkinson's disease, traumatic brain injury. We're learning more and more about the therapeutic benefits of ketosis as an intervention for a variety of ailments.
So tell me how you got started down the path of studying some of these topics along ketosis.
Well, of course, we've worked in the epilepsy field for years, and it was clear to us many years ago that these ketogenic diets had remarkable therapeutic benefit for managing seizures, initially in children. Now it's moving a little bit more into the adult population. But for refractory seizures in children, the ketogenic diet is still recognized as one of the most effective ways to manage difficult to control seizures. And we developed some of the most natural animal models of epilepsy. And we were really amazed at how powerful this therapy was in managing seizures in these mice that replicated the full condition in humans. And then we had been doing a lot of work on the biochemistry of cancer. And we realized that by restricting calories, you lower glucose and elevate ketones. The same things that happen when you use ketogenic diets to manage seizures. And we were surprised at how powerful the diet was in controlling cancer growth.
So let me back up for a second. That's to say that even if you don't really change the macronutrient ratio in your diet, simply by having less total calories, that takes you in the ketotic direction just by virtue of that alone?
Well, it can, but the most powerful way to do that, in humans at least, is therapeutic fasting, which is water-only fasting. That brings the body into a state of ketosis, what we call therapeutic ketosis, within about 3 to 4 days, depending on your age and health status. So just drinking water or green tea or something like this, no macronutrients, can bring you into a state of ketosis within 3 to 4 days in the human. And of course, humans can fast for significant periods of time, upwards of a month or longer, remaining in a very healthy state as long as fluids are present. So this brings us into what we call therapeutic fasting, and it's really therapeutic ketosis, which is remarkably healthy to the body.
The average person, you pick sort of a healthy adult human off the street, and water only, they can really remain healthy for a full month?
Or longer, depending on how much their body weight is initially, because George Cahill, who was the director of the Joslin Diabetes Center here in Boston, he was the leader on starvation in man, and he would test these approaches on people who were obese and diabetic and a variety of different ailments. And he was able to fast some people for as long as 6 months, depending on their body weight, and remain perfectly healthy as long as their fluid ionic balances were in order. So thin people might be able to fast for 3 weeks, whereas obese people could fast for months. The body stores the energy, and you just release the energy from the storage, the fat. And the fat contains a number of fat-soluble vitamins. The bones contain minerals, the liver contains vitamins, so you really don't need much vitamin supplementation. Unless you're going for a longer-term fast, say, over 3 weeks, you might need some supplementary vitamins. But for 3 weeks, you don't need it.
How low are blood sugar levels dropping? Does that mean your blood sugar is at zero by that point, or just at a point where that sort of metabolic switchover happens?
No, your blood sugar will never go to zero unless you use insulin or some other kinds of drugs on top of that, because the liver will make glucose. Let's put it this way: it'll make glucose from the backbone of the triglyceride, the fatty acid triglycerides. When you metabolize triglycerides from storage fat, the glycerol backbone of the fat two glycerol backbones can be conjugated in the liver to make glucose through gluconeogenesis. Also, certain amino acids, the amino groups can be removed and the amino acid backbone can be conjugated into glucose. So this is a process of gluconeogenesis. And when you stop eating, the body then starts making glucose from within. So your blood sugar definitely goes down, but it doesn't go down into pathological states. And the other issue, of course, is that as the ketones go higher from ketosis, blood sugars can go down even further. And, we've experimented with this. And my colleague, Dominic D'Agostino at the University of South Florida, was able to get his blood sugar down to 25 milligrams per deciliter, which most physicians would consider a catastrophic state. He's perfectly lucid because his blood ketones are elevated. So ketones replace glucose for the brain, and you don't go unconscious, and this now becomes a non-issue.
You published a book recently on cancer as being a metabolic disease, which is a very new way of thinking about it. Can you tell people what led you down that road and kind of give the overview of what you're theorizing there?
Yeah, well, it became clear that when you do fasting or ketogenic diets, what's the mechanism by which these approaches manage the disease? I mean, the question is, how is it so effective in reducing tumor size, targeting blood vessels, targeting inflammation, targeting all the aspects of cancer that the pharmaceutical field and the academic field are trying to develop drugs for? Yet this simple procedure can do the same thing in a more effective and less toxic way. So the question is, how does all this happen? And, years ago, Otto Warburg said the underlying cause of all cancer is insufficient respiration, and therefore the cells must ferment. If the cells cannot respire like our normal cells can, in order for the cell to remain alive, it must have an alternative form of energy, which is the primitive form of fermentation, which is a process of energy formation that existed for organisms on the planet before oxygen came into the environment. So our cells have this capacity, but it takes a while to transition from respiration to fermentation. And what happens in cancer is many aspects of the environment damage respiration of cells gradually, not acutely, but gradually or chronically. And this then gives some of those cells an opportunity to reconfigure fermentation. And fermentation, then, the cells become fermentative. And all cancer cells are fermenters in one form or another. So then you say, what is the fuel for fermentation? It turns out to be glucose and certain amino acids. And these diets target the glucose and these amino acids, and therefore it targets the cancer naturally.
Interesting. So you're kind of starving out the cancer's natural energy supply.
Yeah, you're putting pressure on it. This is only the first step. It makes the cancer in a much more indolent state and makes the cancer cells extremely vulnerable to other kinds of non-toxic drugs. And then, of course, this is interesting in the sense of how to target and manage cancer and also how to prevent cancer. But the issue, what the field is doing, is they're spending enormous amounts of time and energy looking for personalized therapies based on genomic defects in the tumor cells. And this is the main thrust of the entire cancer field, whether it's in academic, in hospitals and research centers, or whether it's in the pharmaceutical industry. The issue then becomes, is cancer a metabolic disease or is it a genetic disease? Now, when you look at the research from the past, I've bundled up a number of publications that were scattered throughout the literature over 40 years, showing that if you take the nucleus of the tumor cell and put it into a normal cytoplasm containing normal mitochondria, the cells no longer behave abnormally. They grow normally. So the key feature of cancer cells, which is abnormal growth, can be completely corrected when you take the nucleus of the tumor cell and put it into a normal cytoplasm. This has been shown by the top labs in the world over a 40-year period. People just simply ignored those findings. So what it means is all those genetic mutations that you see in the nucleus and that everybody is focusing on are really insignificant. They're downstream epiphenomena. Yet the entire field is focusing on something that's not directly relevant to the disease. And this is a major problem. But you can make tremendous amounts of money targeting genes, which really are not the central issue of the problem. It's much easier to target the fuel that the cancer cells use than in targeting the thousands and millions of gene mutations that have been documented in these tumor cells. So the whole thing is upside down. And that's the reason why we've made no major progress in the battle against cancer.
You're saying there's 40 years of evidence with studies showing that essentially it's not the cellular DNA that's the key point here with cells turning cancerous. What do people say to that? Like when their studies are focused around that and kind of ignoring this, what seems like a fairly crucial piece of evidence, what is the retort?
That's a very important point. People say, well, this is real. How come nobody has recognized it? And it's very interesting because those papers came out sporadically over a 40-year period as anecdotal contrary observations to the gene theory of cancer. And they were more or less discounted as kind of oddball kind of observations. The issue, of course, is they were done in the top labs in the world. They were done by the leaders in the field. But they came out sporadically, like one paper, 1 year, and then 5 years later, another paper. And they were in different kinds of journals. And what I did in my book was bundle all those findings together and present them as a unified whole, with all the findings pointing to the same general conclusion, that the nuclear DNA is not the driver of the disease. It's only when you look at all the studies combined together, one after the other, all done independently of each other, using different organisms, human cells, mouse cells, frog cells, cloning mice from tumor cell nucleus, and then you still see the so-called driver mutations in the normal tissues of the mouse cloned from the tumor. You have to say, what is going on here? I'm the only one who's put those things together. And now the field seems to be in a stunned state of silence. They don't know what to say about it because I've got there's no blowback. And I know people are reading it, and I don't think they have an answer to this. The answer to this is cancer is not a genetic disease, and that throws a whole stick in the spoke of the entire industry.
So when exactly did you drop this bomb on the scientific community? I just heard about this maybe about 3 weeks ago, but I know that your book came out, what was it, 2012?
Yeah, it was there in 2012, and I put it together in a chapter in my book, and I've since published a couple of articles on it, and people just can't believe it. The ones who believe it, most of the people in the lay community, they seem to understand it. It's amazing. I find it completely remarkable that people without a scientific background can completely understand this phenomenon. But those working in the top labs have huge difficulty, either ignore it or they can't grasp it. The argument is, we need more studies. And the studies were done by the top guys in the field. You can't get any better studies than the ones that are already done. There's nobody else who can do a better job. So I don't know how they're going to deal with it. That's their problem.
What are the credentials like of these people who did the original studies? I mean, are they jumping to your aid and sort of on the bandwagon with what you're proposing as sort of the follow-on effect for this?
Some of the top labs, like Dr. Yanish down at MIT and Beatrice Mintz, these are giants in the field. These people, Jim Morgan at St. Jude's Hospital, Shea and Werblin, people say, we should do it again. Yeah, well, if you can do it better than the giants in the field, that's great. But they've already done it. It's just that what the conclusions from their experiments was not that cancer was not a genetic disease. It was cancer was some sort of an epigenetic phenomenon that didn't directly involve the nuclear genome, some reprogramming event. But they didn't understand that Warburg had clearly defined many years ago that cancer was a mitochondrial disease. It's a mitochondrial metabolic disease, with the exception of one paper by Ruth Sager's group. Unfortunately, Dr. Sager passed away of cancer, but she was a leader in the field, giant in the field. She was the only one that recognized that these findings are linking to the metabolic theory of cancer that Warburg had defined many years ago. So Warburg was, as they say, thrown under the bus. He was the number one biochemist for the 20th century and won the Nobel Prize for his work on respiration in the 1930s, a giant in the field himself. But I think the addiction of the field to the gene, the DNA structure and function and all this kind of stuff, the field just, with a herd mentality, ran off chasing genes and forgot all about the essential biochemical aspects of the disease.
Is there a pharmaceutical industry angle to this where the ketogenic approach is so relatively inexpensive that they kind of don't want that to be the solution?
Well, I don't think they don't want it. I think it's one of how can we make profit on it? I think that's been mostly recognized by the food industry. I think the food industry is coming to recognize this as a potential to generate revenue. If you go address this, what I just said to you, to the pharmaceutical industry, they mostly stand in stunned silence. They don't know what to say about it. People have to come to realize and know these findings. They have to sit down carefully and evaluate the data, and then they have to come to their conclusions. Right now, the main thrust in cancer is immunotherapies. These are antibodies that are made to certain protein defects from the Genome Project. So they're going to treat these people with these antibodies, and you'll see in the Wall Street Journal, a number of other publications, how these cancer survivors and how healthy they look. But you have to read between the lines and see how many people are dying from these immunotherapies. And you have to see the toxic effects and the exorbitant cost of using immunotherapy to treat cancer. I mean, it'll work on some people. There's no question about it. But the majority of people don't respond, or they have a very partial response, and it costs them a fortune. They have all these toxic effects. And you might be able to avoid all that if you recognize this is a metabolic disease. You don't really need to do a lot of that. Not to say that that is not important. We like to shrink the tumor down using metabolic therapies, and then possibly these immunotherapies could be used as a final solution to the problem, not the primary solution.
Can you give some bullet points of the reduction in tumor size and tumor count and things like that you've seen in your mouse studies?
Yeah, so this is a very important point. Most people who look at calorie restriction in cancer see major effects in a variety of different ways. You could shrink down the tumors significantly in size. You can reduce blood vessels. You can significantly reduce the inflammation and the signaling pathways associated with that, which are all part of trying to manage the disease. And you can do it in a non-toxic way.
What would be the dream study that you'd love to put together?
The problem with the dream study is that it conflicts with the standard of care, which is what the units in the hospitals do. Let me give you an example. When you have brain cancer, you do the standard of care, which is generally surgical debulking. You remove as much tumor surgically as possible. And then the patient is given a regimen of high-dose radiation therapy to target the tumor cells. And then either the radiation is done first, followed by temozolomide, which is a toxic alkylating agent, chemotherapy orally administered. And it can be done either in conjunction with radiation or following radiation. And then this is the standard of care. And this is what appears to be written in granite. It shouldn't be, but it is. And it's very hard to get away from that. Now, those standards of care themselves, I've published a big paper showing that they're actually counterproductive to the long-term survival of the patients. They create a microenvironment that guarantees the demise of the patient. Now, occasionally you'll get somebody that survives for a longer than expected, but you get 5-year survival of less than 1% or about 1% for GBM, glioblastoma, and then they give high-dose steroids. All of the standards of care for treating cancer are counterproductive to the long-term survival. They increase blood sugar, they increase the availability of glutamine, they increase all of the micronutrients that these tumor cells will need to survive and recur. So this makes no sense. Now, this is a hard pill to swallow because this is what they do everywhere in the Western world. They treat people the same way. If you're in India, Germany, Japan, no matter where you are, you're going to get the same treatment, which is what I just told you. Now, if you're going to do metabolic therapy, You have to do it as an alternative to the standard of care. Now, you can sometimes dovetail radiation with ketogenic diet, as Adrian Scheck's group has shown out at the Barrows Neurological Center. So there's a possibility that radiation could work synergistically with ketogenic diets, but I'm still working on that. But the best way to do it, in my view, is patients would be debulked. That means they would have the surgery. That's a necessary, because that's been shown that the less the tumor, the longer the person will live. And then the metabolic therapy comes in and shrinks this tumor down more, creates a more indolent environment. And then there's add-on therapies like hyperbaric oxygen. There's various drugs that work together with calorie-restricted ketogenic diets, or what we call therapeutic ketosis, that will manage these tumors in a less inflamed environment, a less provocative way. This is the way it should be done. But unfortunately, it's very difficult to get that done because it has to reject the standard of care. And that's a big issue right now.
I remember hearing more than once that when people have radical cancer therapies that they can't do for whatever reason in the U.S., that a lot of times people will go down to Mexico and will sort of have an alternative therapy in a supervised environment in Mexico. Is that something that still happens, or has that gone the way of the dodo?
No, I think it still happens. Like sometimes people will use a Collie approach, the Collie vaccine, which underlies the William Collie years ago treated people with live bacteria, and they would get horrific fevers, and oftentimes if the patients survived the fever, a lot of times they would have a complete cure to the cancer. Now, sometimes you can do that in Mexico. I don't know if they still allow some of these things. A lot of people will use inactivated bacteria, but still induce high fevers in patients. And the high fevers, it's a form of metabolic stress on the tumor cells. So again, the tumor cells are vulnerable to metabolic stress. Very high fevers put tremendous stress on tumor cells. Sometimes this is done in Mexico, sometimes it's not. So it depends on what you're capable of doing under the medical profession. But it's very interesting to note that the major thrust of the pharmaceutical industry is in what they call immunotherapies. There's a number of papers showing that immunotherapies work best in those patients that get high fevers. So, what's working here? The $100,000 drug or the high fever? And this is the same thing with calorie restriction. A lot of these cancer drugs cause patients to get nauseated, vomiting, fatigue, all this kind of stuff. These are indirect forms of calorie restriction. So one never ferrets out, what's the why are people responding to these drugs? Is it because they're killing tumor cells through a very toxic mechanism, or is it killing tumor cells because the patient's not eating, he's so sick?
Tell me about that a little bit, because I know that one of the differences between mice and people is that the metabolic rate of the mouse is just off the charts compared to a human. And so some things like amount of food intake per kilogram we can't just multiply mouse weight by human weight and figure that out.
That's a really important issue. The basal metabolic rate of the mouse is like 8 times that of a human. A mouse will starve to death in 5 days if you take the food away from it. As I said, depending on the body weight. And the same with the mouse too. The heavier mice can live longer without food than thinner mice. And the heavier humans live longer without food. But a normal sized 28-year-old man weighing about 180 pounds can probably survive 70 days to 80 days on water alone before dying of starvation. But a mouse will die in starvation in 5 days. The mouse's heart rate is like 600 beats a minute. If a human had that, his head would blow off. So you're absolutely right about this. Now, here's another interesting thing. One of the top cancer models that they use in the field is the patient-derived xenograft. So they take a piece of tissue, cancer tissue from a human, and they put it in the body of this immunocompromised mouse to grow a tumor. And then the tumor, of course, grows in the mouse's body, the human tumor growing in the mouse's body. And they take the tissue out, and they do a genomic screen to find out if there's some gene that they can target, and they go back to the human. It takes a long time for the tissue to grow in the mouse. Many times the human that donated the tissue dies before the mouse develops a tumor. Then the mouse develops this human tumor growing in this totally different metabolic environment. The human cells start taking up all these mouse proteins and lipids, and now it becomes some sort of a centaur hybrid kind of thing. It screws up the genomic thing, and they're spending millions and millions of dollars on these absurd models. It's comical but tragic at the same time. And they don't understand the differences in the metabolic rates, or they don't care about them. And then people wonder why none of this stuff works. I mean, to me, it's very clear why none of this works.
What are some of the practical applications, both for somebody who maybe has cancer now, or for just everybody else who doesn't have cancer but is worried that cancer happens in enough people that it's something that we should all be worried about?
Prevention is the most important thing, but most people throw caution to the wind, and we all think, none of us are ever going to get cancer until we get it. And then once you have it, what are you going to do for me? So the question is, what are you going to do? Right now, the treatments for cancer are quite toxic, and I think everybody recognizes this. Now, not everybody will experience tremendous toxicity from whatever therapies that they might have for whatever type of cancer they have. But I think a lot of people fear the treatment as much as they do the disease. They don't want their hair to fall out. They don't want to feel sick and fatigued or nauseated. They don't want all of the pain and suffering that goes in with the treatment. Yet they do that because they feel that this is going to cure them. And many times it does. Don't think that this standards of care don't work. I mean, there's many people who achieve remission, prolonged remission. The problem, of course, is that your body takes a beating oftentimes as the result. You have now subjected your body, or your body has been subjected to, a series of toxic therapies in one form or another. Now, everybody knows radiation is not good. Radiation causes cancer, yet it's used to treat cancer. But the radiation itself will damage the tissue architecture of your body. So you're left with some sort of a remembrance of this treatment. And if you take toxic drugs, you can get chemo brain, you can get all kinds of problems in digestion and other things that will linger with you for years. But people will say, well, I'll accept that because I'm still alive. And therefore, I dodged this bullet, but I have this toxicity in my body as a reminder of my survival from this disease. My view is that if you treat the disease as a metabolic disease, where the therapies are far less toxic and potentially equally as effective, that seems to be the way to go. And therefore, the patient emerges in a healthier state after managing the disease rather than in a less healthier state. Because these less healthier states eventually take years off your life sometime down the road. So the question is, can you manage this disease in a way that doesn't induce toxicity? That's the question. And metabolic therapies offer that opportunity. It's just that they're very underexplored and underutilized.
So when I first heard about what you were studying, it was actually involved with prevention and how somebody might be able to prevent ever getting cancer in the first place. If you have a couple of stray cancer cells somewhere in your body, how you could use a ketogenic fast to essentially starve them out. Can you talk a little about that?
If we understand the origin of the disease, and this goes back to cancer as a metabolic disease, not a genetic disease. If we understand that the origin of the disease is damage to the respiration of the cell, which is basically the mitochondria, it's a little organelle in our cells that generates energy efficiently. So when we breathe in oxygen and we expel CO2 in our breath, this is the evidence that our cells are respiring. And healthy respiring cells do not get cancer. Cancer comes only to those cells that have insufficient respiration. This was Otto Warburg's contribution, and we have expanded upon that and shown that. So to prevent cancer, you have to keep your cells in a high state of efficient respiration. It's that simple. Okay, how do you do that? Well, you can do therapeutic fasting, which lowers blood sugar and elevates ketones. When the ketones come into the cell, they generate more energy, the carbon-hydrogen bond, without generating reactive oxygen species, as would be the metabolism of glucose, fuels derived from glucose. So ketones are what we call super fuel. They enhance the energy of the mitochondria and make it healthy, and respiration becomes more energetically efficient when the cells are burning ketones. How do you get ketones? You have to lower the glucose and you have to elevate these ketones. This will protect your cells against the formation of a tumor, which comes in cells that have inefficient respiration. So the way to prevent cancer is simply keep your mitochondria healthy. And how do you do that? Well, you avoid the situations that will damage respiration. You avoid carcinogens. You avoid, viral infections can cause cancer. All of these things damage respiration. So the way you can do that is you therapeutically fast, or you periodically take a ketogenic diet for 4 to 5 days or a week. All of these things can enhance the energy efficiency of your respiration. You can exercise, you can lift weights, you can do all of these things will give you energy efficiency. And that will protect your cells from getting cancer in the first place. But, a lot of people don't do that. And we're also bombarded with a lot of poorly nutritious foods. They're empty calories. They don't have the nutrients that will enhance mitochondrial respiration. They're empty calories. They provide a lot of glucose, but they don't provide the kinds of nutrition that would help strengthen our mitochondria. So, these are personal choices. But I think more and more people are becoming aware of these facts, and hopefully things will change.
So tell me how you've applied these ideas to your own diet, your family's diet, things like that. Do you do a ketogenic fast every now and then?
I've done the 3-day fast. And of course we do ketogenic diets occasionally, but I haven't done it long term yet. But if I had cancer, I certainly would do it. The issue is, you know what to do when you have to do it. It's like having the spare tire in the car. If you get the flat, you're going to put the spare tire on there. And I have to admit, not eating food for 3 days and just drinking water, it's doable, but as my students say, it's uncomfortable. You don't feel at the top of your game. But some of my students who've done it longer term have actually found it to be not so debilitating. And I think the younger you are, the easier it is to do. That's another thing.
Do you think that there is a prophylactic, preventative type fast? If you had a healthy 20-year-old and they're like, you know what, I just don't want to get cancer ever. What can I do every 6 months, every year, whatever it is, to take these ideas and turn them into a practical application that I can use to eventually immunize myself to cancer?
Yeah, well, I think that's what they would, as I said in my book, if you could possibly fast for 1 week out of a year. If one could do a 7-day fast, and I know this, believe me, it's not like, oh yeah, everybody's going to run out and do this. No, they're not. Believe me. What the body does is it starts to cannibalize. I call it autolytic cannibalism. What it does is starts to look for cells that are not functioning at their maximum. And those cells are now consumed by other cells, macrophages and these kinds of cells. They're kind of like monitors and patrollers of the body, and they eliminate inefficient sources of cells and things like that. And those cells are digested and their nutrients are then recirculated in the bloodstream, and these nutrients now go to other cells in the body that have efficient respiration. It's an efficient way to so-called cleanse. They always like to use the word cleanse. What are you cleaning, basically? You're cleaning up dead cells, inefficient cells, and in this way, the ones that are left are at a higher metabolic efficiency. And if the cells are at a higher metabolic efficiency, they're less prone to become cancerous. Let's put it that way. The risk of cancer would go down if you could eliminate cells that have inefficient respiration. And therapeutic fasting brings the body into that state, and that will prevent cancer. Now, I can tell you anecdotally, I've spoken to the people of the Calorie Restriction Society. There's a group of people here we have in the United States and around the world, actually, that practice this on a regular basis. They eat very few calories. They're all very thin. They're all very healthy. And I asked them, what's the incidence of cancer in your group? And they said it's extremely low. They could only remember 1 or 2 people ever in their group ever getting cancer. So these people are in this constant state of calorie restriction. And the incidence of cancer is lower in their group than it is in the general population. So, this has not been evaluated by epidemiologists, but it should be. There are these people. What are the incidence of cancer in these people? Compare it with the general population, and you begin to see clear differences. So, this supports the idea that if you are calorie restricted, your body is already going through this therapeutic surveillance, and the probability of the risk of cancer is going to be less. It's just that simple.
Is there anything else that you didn't get a chance that you'd like to circle back to, or anything else you'd like to mention?
I just think that the population has to come to understand and appreciate what our bodies are capable of doing. Our bodies can heal themselves in many different ways if they're given the opportunity to do that.
Smart Drug Smarts.
So thank you very much to Dr. Seyfried for taking the time to join us for that interview. I think there's a ton of really interesting information in there. And of course, like everybody that, as far as I know, I don't have cancer, but I'm super glad about that. But it's nice to know that there's possibly some growing tools in the arsenal against cancer. And in this case, sort of a natural and accessible non-pharmaceutical one, which is always a couple of nice feathers in the cap. I hope that you guys don't mind my sort of indulging myself in doing an episode that wasn't terribly on topic for smart drug stuff, but I just can't help but feel that general health and physiology does sort of have a place, since all of that's going to factor into a person's cognition. But yeah, if you're violently opposed to or radically in favor of some of these sort of digression episodes, I would love to hear your opinion either way and just sort of take a little taste test of the audience and see what people feel about stuff like this. But moving right along, let's do the ruthless listener retention gimmick.
Smart Drug Smarts. Ruthless listener retention gimmick.
As we settle into Christmas season, there's a lot of seasonal drinks and probably other recipes too. I'm not much of a cook, so I don't know all the places it's used, but nutmeg. Nutmeg is something that we always think of as being sort of a holiday food. Nutmeg is actually the seed of an evergreen tree, Myristica fragrans, which is native to Indonesia, although it's now cultivated all over the place. And the spice comes from a thin protective layer that encloses the seed. Well, apparently nutmeg, in addition to being like a tasty spice, at not terribly large quantities can start having some psychoactive effects and toxic effects as well. In the Middle Ages, nutmeg was actually used to make women sort of go into spontaneous abortions. If a woman wanted to get rid of a baby, then intentionally sort of overdosing on nutmeg was at least a sometimes effective way to do it, although not a very pleasant one. And more recently, prisoners in the penal system have used it as a sort of miserable drug substitute. In the autobiography of Malcolm X, he talks about purchasing nutmegs from inmates in prison. It was concealed in matchboxes and stirred it into water, and that a penny matchbox full of nutmeg had the kick of 3 or 4 reefers, according to Malcolm X. But toxicologists say that if you're looking to get high, probably nutmeg's about the worst possible choice you could make, because despite having some psychoactive effects, including sort of an out-of-body sensation and sometimes even visual hallucinations, more common are intense nausea, dizziness, extreme dry mouth, and a lingering slowdown of normal brain functions, says Dr. Edward Boyer, a professor of emergency medicine at the University of Massachusetts Medical School. People have told me that it feels like you are encased in mud. You're not exactly comatose, but you feel really sluggish, and your remembrance of events during this time period is incomplete at best. So the takeaway from this, I guess, is just watch the amount of nutmeg that you're adding when you're throwing in a dash of this and a pinch of that. Just make sure you keep your dashes and pinches of nutmegs to reasonable quantities, because apparently at about the 2 tablespoons threshold, people can start exhibiting some of these toxic/psychoactive symptoms.
Smart Drug Smarts, the podcast so smart, we have smart in our title twice.
You heard it. That is the episode. I'm pretty happy to report that we've actually got, I think, 3 more interviews scheduled before the end of the month. We're trying to jam-pack all these interviews before the official Christmas holiday stuff gets underway and nobody wants to jump on an interview phone call with me. But yeah, it should be some good stuff coming up in the later part of December. So stock up your iPhone with these podcast episodes as you're doing your Christmas traveling. I'm actually about to jump on an airplane myself in about 6 hours. So it's starting to get to be that time of year. If you are feeling particularly giving during your holiday seasons and you want to throw a bone to your friends at Smart Drug Smarts, it is always a giant help if you wanted to jump over to iTunes or Stitcher, whichever platform you prefer to do your podcast listening on, and leave us a positive, glowing review. Will help more people find out about the podcast and be very much appreciated by yours truly and the entire Smart Drug Smarts family, including Mimi the cat. The show notes for this episode will be online at smartdrugsmarts.com, including the links to everything that we talked about here, and I'll be back at you in a week or less with the same unflagging commitment to helping you fine-tune the performance of your own brain. Have a great week and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.